All right, folks, welcome back. Module 18 in Unit 3. We're going to talk about all about vision, light energy, as omitted in light wavelengths. We're going to talk about parts of the eye and how they function. You will need to know those parts of the eye and how they function. So please make sure you pay attention and don't hesitate to ask questions. Okay, so in this module, we're going to discuss the characteristics of what we call the visible spectrum. Okay, we're going to describe how the structures in the eye work and how they help to direct, interpret, and then send on for processing all of those energies. We're going to talk about rods and cones. We're going to talk about color vision. We're going to talk about how color is important. We're going to talk about feature detectors. We'll also explain the use of parallel processing and how it helps us create visual perceptions. Okay, here we go. All right, so unfortunately, us humans, if you look at this diagram here, us humans can only interpret a fraction of what we know to be the wavelength of lights, various lights throughout that exist in our world as far as we can tell. Okay, on the far left-hand side, you'll see we have gamma rays. I got a little cut off there. Then X-rays, this is what we use to take scans of bones. Ultraviolet rays, this is what is emitted by the sun that can burn you, UVA, UVB. Then we have this little rainbow here. Notice the rainbow mirrors the colors on either side. So what we're going to do is we're going to go from ultraviolet, where we can't see, to violet that we can see. And then it's the rainbow in reverse. Okay, so purple, blue, green, yellow, orange, red. Okay, infrared. Infrared is basically beyond the spectrum of normal vision. All right, red waves are very, very slow, whereas ultraviolet are incredibly fast and tight. Okay, all of those waves, if you can remember the wavelength, it goes from slow to the right. Okay, these are very slow wavelengths on the red side. These are very fast in terms of frequency. Okay, we'll take a look at those frequencies in the next slide. All right, so frequency is basically the wavelength, the length from trough to trough or from peak to peak. This is peak to peak. This is trough to trough. Since they are wavelengths, they are symmetrical on both the top and the bottom if you were to draw a center line. Notice blue, again, like we said, ultraviolet, blue, all right? Blue has a very tight, wavelength, all right? A very focused wavelength. Red, opposite of blue, is a very long and drawn out wavelength, okay? Now let's talk amplitude. Amplitude is the height of those waves, okay? Blue and red we use to describe the length of the wavelength or the lack thereof. Blue is very short and tight. Red is very long. Now, yellow and green we use to measure the height or lack thereof. Yellow has a very strong amplitude. All right. Green, on the other hand, has a very dull or a very small amplitude, a very low wave height. Wave height is measured from the top of the crest to the bottom of the trough. Okay, pretty easy stuff. All right, so structures of the old eyeball. Now, one thing I want you to draw your attention to immediately is this guy right here, the candle. Now, let's just say we are going to take a close look at the wavelength of light that is coming from the flame. That flame is going to enter the top part of your eyeball. And then what happens is this. It's going to hit this lens and it's going to be inverted.
Buses will be leaving in 5 minutes. Buses are leaving in 5 minutes.
Better catch your bus. Okay, so if you'll notice, the candle is now upside down. And it's actually spun not only top to bottom, but also left to right. So the image is completely inverted, okay? So here are the parts of the eye, starting from outside in. Your cornea. Your cornea is the coating that goes around the lens, okay? the coating that goes around the lens. Your iris is the muscle. That is what gives you your eye the color. All right, your iris is that muscle, and it actually looks kind of like a muscle. If you look really closely into somebody's eyes, make sure you like them before you do that. But when you look really closely at somebody's eyes, you can actually see the muscle fibers in the pupil itself, or I'm sorry, in the iris. The pupil is the void or the lack of covering in the middle of the iris. Okay, the pupil can either expand, or what we call dilate, or it can constrict or contract to a very tight space. We do this to be able to control the light that is allowed through into the lens and onto the back of the retina, because if you always have heard, don't look at the sun, it's too bright. That is true. All right, UVA, UVB light can come into your eye and damage the back of your retina. Okay, not only will you have temporary sunspots, you can also cause irreparable damage. Okay, so the lens again, this inverts and flips the light rays coming into your eyeball. Okay, and that projects on the back of your retina. The fovea is just above your blind spot. Okay, the fovea is, for human beings, basically straight back through the eyeball. Okay, and this is where most of the light energy is going to hit our retina in the back. Okay, so the retina is basically the back part of the eyeball, and it goes pretty much all the way around. If you'll notice here, you can see this is the retina. Okay, so then we have the optic nerve. The optic nerve is basically a channel and a bundle of different types of cells, and those cells are going to interpret all of this data from the back of the retina, and it's going to be piped from the retina into the optic nerve, which goes into the optic chiasm, which goes to the visual cortex. And depending on whether this is the right eye, right visual field, left eye, left visual field, et cetera, it's going to go to the different hemispheres, but still go all the way back to the visual cortex. Okay, there is a blind spot in all of our eyes. We will do an activity in class where we will try to identify and find your blind spot. Don't worry, we all have it. Perfectly normal. Okay, so the cornea, outer cover. We've already talked about that. Basically, it helps protect the eye. It keeps the eye moist. And it basically is the first entry point for light. Pupil, like I said, is that center point where light comes through. It is adjusted by the muscle tissue called the iris. The lens, again, inverts that light and focuses it back to the retina. Please keep in mind that when that light is hit, when it strikes the back of the retina, that image has been inverted upside down and right to left. Okay, so how does our lens change shape? Well, the lens can actually change its size. Okay, it can actually adjust its curvature, which changes the intensity of the projection of that image on the back of the retina. Okay, and it allows us to focus on things that are very close or things that are very far away. So if you focus on something on a wall, say a good 10 or 12 feet or farther away, focus on that, and then keep focusing and put your finger up in front of your face. You can't see any detail on your finger. Now shift your vision to your finger. Your finger needs to be about 6 to 12 inches away from your face. Okay, so that being said, people that suffer from nearsightedness, they suffer from something called myopia. This is dealt with by either surgery or corrective lenses. Corrective lenses can be glasses or contacts. Hyperopia, or farsightedness, is the fact that the lens focuses light past the retina, and this is much harder to cure because it has a tendency to often over adjust, and glasses and contacts can help, but it's a little more challenging to correct farsightedness as opposed to nearsightedness. Okay, again, what is the retina? The retina is photosensitive or light sensitive. It's the inner part of the eye, and it contains rods and cones. Rods and cones work together to interpret the different types of lights that come into the back of our eyeball. And these are broken into shades of black and white or color, depending on what it is we're looking at. Okay. So again, the retina is pretty much the back half of the entire eyeball, and it's almost spherical in shape. But let's take a look at what happens in the retina itself. All right, if you take a look here, so what we have rods and cones in the back of the retina. Okay, and what happens is this: so light comes in, and light actually will make it through the outer layer of your retina. All right. And what happens is that it's going to trigger ganglion and bipolar cells. Okay, here are your rods and your cones. If you notice, an easy way to remember rods and cones and what does what, just remember cones are color, and if you remember that, rods will be black and white. Okay, or grayscale. Okay, so what happens is this: as that light comes in, the rods and the cones are going to generate an outgoing sensory or neural impulse. Okay, and that neural impulse will be pushed right back out and then down the layer, the inner layer of the retina, and onto the optic nerve and into the optic chiasm. Okay, that's basically how it works. All right, so what are rods? Rods detect black and white and gray. They are highly sensitive to movement, and they are best used at night. Okay, rods are located along the outer periphery of the eyeball. And what I mean by that is this: if you look right here, these are the rods, these green looking things. All right, they're very close to the outer periphery. And we have to have rods when we're dealing in twilight vision. All right, twilight, like dawn and dusk, our cones do not handle that in between time very well, nor do the cones handle darkness very well. So the rods really take over at night, which makes sense because we don't see a lot of color at night anyway, because our eyes aren't that good at night vision. Okay. Basically, they share connections with a single bipolar cell, and they send a combined message to the brain. We have faint light and peripheral motion. All right, that's what the rods are really good at, like motion off to our left and our right eyeballs, maybe to the top and to the bottom. Our cones, on the other hand, excuse me, our cones are going to be photoreceptors, and they are going to be near the center of the retina, and they are very well functioning during the day or well lit conditions. Okay, they're very good at fine detail, and they obviously give us color sensations for those folks that are not colorblind. So what we see is cones are clustered in and around the fovea, which makes sense. The fovea is the focal point for a majority of the light coming into our eyeball and hitting the back of our retina. So having a cluster in and around the fovea it makes sense. Dim light, the problem is the cones shut off because they can't really interpret the color. All right, the wavelengths get blurred. Okay, a lot of cones have direct connection to the brain. Basically, a single nerve wire heading from the cone and into the wall of the retina, and then in through the optic nerve and into the chiasm and straight into the brain. Not all of them, but a lot of them do. Okay. All right. Most cones operate on a single bipolar cell. Bipolar means it transmits both ways. Okay. All right. The fovea. The fovea is the focal point around which the cones cluster. This gives us our greatest focal point, meaning we're most able to focus when light hits right here. So when we're looking at something we're trying to focus, we don't tilt our head up or down to try to focus usually. Now we might if we have bifocals and we're trying to use the lenses, but that doesn't, we're trying to use our eyeballs to focus, and usually that focal point is straight ahead. All right. The optic nerve. The optic nerve is the gateway that carries all of that neural data, the sensory data from the eyeball to the visual cortex. Okay. It's pretty much one way. All right. The blind spot. Y'all have a blind spot. Okay. A blind spot is created by the optic nerve. All right. There are no rods or cones there because it's an entry point. It's where all the nerves connect, and it's where they all go into the optic chiasm. Okay. The chiasm being like channel. Okay. So what happens to the neural impulse after it exits the eye? We talked about this a little bit when we talked about um, the hemispheres of the brain. If you'll notice, left eyeball, left hemisphere goes, I'm sorry, left part of the eyeball, left eye goes to the left hemisphere. Left part of the right eye goes to the left hemisphere. The right part of the left eye goes here, all right, to the right side, and the right part of the right eye also goes to the right side, all right. This is the visual area of the thalamus. Keep in mind that the thalamus is responsible for channeling all of our sensory data. Then it's piped into our visual cortex in our occipital lobe. Okay? All right. So, two theories we need to know. Young Helmholtz trichromatic or three-color theory. Basically what this says is we have three color receptors, RGB, red, green, blue, okay? And basically, each one of these cones are stimulated by the wavelength in a certain frequency, or a certain combination, I should say, kind of like a decoder pen. All right, so once we hit a certain combination of blue rods, red rods, green rods, it's going to create a color, or a perception of a color at least. Okay, now again, keep in mind this is a theory. We don't really know 100%. Okay, so people that don't have that are colorblind basically will look at something like a football game and see this. Okay, so if you don't have color vision, all right, you will see this image on the left right here. The image on the right is a normal person's vision without color blindness. Now, the reason we think the young Helmholtz trichromatic theory might be correct is because of color blindness. All right, and the idea is that let's just take these jerseys here. So we have green and we have red. If you look at the jerseys over here, there's no such thing. Okay, it's all one kind of dark green gray color, kind of like blah. Okay, so what are some characteristics of color blindness? 1 in 50 people is colorblind in average. Males are usually more affected. It is genetically passed, usually from generation to generation. Not all people have full color blindness, which is red, green, blue. Basically, it's a combination. A lot of people have, like, say, red green blindness, or they'll have blue yellow blindness. They won't have all blindness. Vision is monochromatic or dichromatic. Make sure you know these two terms. Mono meaning one, chromatic meaning color, dichromatic, two color. Okay. All right, let's talk about the Herring opponent process theory. This theory is kind of like each little photoreceptor, each little cone has a tug of war. All right, and the idea is this. Basically, you have a pair of receptors and they're paired with another color, red and green, blue and yellow, white and black. Okay, and basically, depending on the wavelength coming in and hitting that photoreceptor, one of the two colors will win, but not both. So if we have light coming in and hitting that blue-yellow receptor, if it is more of a blue hue, blue will win, and it will send a blue message. If it's yellow, it'll send a yellow message, if that makes sense. So basically the idea is that we have three tugs of war going on in these three different color categories, and every single cone and rod is basically, well, cones are basically either one color or they're the other. And then however much of that color is generated by the neural impulse will be the color that we perceive. Okay. All right. Moving on. So, how does color processing occur? The young Helmholtz trichromatic theory suggests that we have varying degrees of color because of the varying degrees of combination of activation between red, green, and blue cones. So basically, this theory says is that it's about the amount of wavelength coming in. If is it more blue, is it green, is it red, is it a combination? Um, and it really boils down to how many cones of each color, red, green, blue, how many cones get activated. The opponent process theory on the other side basically is all about the response of those cones. Is it going to be a red or a green color? Is it going to be a blue or a yellow? Or is it going to be a black or a white? And any one of those different combinations, you'll basically take the total of all of those cones added up, and that will be the color that the brain perceives. I know that's kind of confusing, but make sure you know those two theories. I don't know if I have any other videos in addition, but anyway, let's talk about feature detectors, the last part of this module. Feature detectors are basically nerve cells located in the visual cortex, and they are going to look at edges, lines, angles, and movements. Why are those important? Well, feature detectors are going to allow us to gain information, and they're going to allow us to basically pass elements of what it is we're looking at through sensory data, the images in our retina, and it's going to allow us to pass those images back to our supercell clusters. Supercell clusters are basically cells that allow us to interpret more complex patterns. Okay, and out of this is where we get things like gestalts and higher level interpretation of sensory data. All right, so let's talk about parallel processing. Parallel processing is a lot like dual processing. Dual processing is where our minds can cognitively hear the sound of my voice and hopefully learn at least a little bit about AP psychology. And at the same time, your body is still digesting its food from lunch. You are either still energized from your rest last night, or you're producing growth hormone, whatever. All right, it's our body's ability to do two things at once. Parallel processing is pretty much exclusively in the field of vision. All right. So what it does is it takes multiple different things, and it looks and interprets multiple different items. So when it comes to vision, this is what we look at: motion, form, depth, color. All right. So parallel processing is when we look at each one of these different things, but we're doing it simultaneous with the other. All right. So as the bird flies through the air, we can see the form. Yes, it is a bird. Yes, it is in motion. Yes, it's not going to run into my face. It's a certain distance away, so we can roughly judge the size. Is it a big bird? Is it a baby bird? We don't know. If we know the depth, we can at least make a guess. And then the color of the bird. Is it male? Is it female? Is it bright? Is it dark? Etc., etc. Okay, that is dual processing. Make sure you know motion, form, depth, and color are all aspects of visual dual processing. Okay, so supercells again. Let's kind of touch base again. So to recognize a face, the brain is going to integrate information projected by your retinas to several different visual cortex areas, and it's going to compare it with stored information. All right. Basically, we call this fusiform. So what we're doing is we're taking pieces and parts. It's almost like Wheel of Fortune, where you get like one letter, then the next letter, and then the next letter, and eventually you can solve it. Some of us, it takes more letters. But when we're looking at a face, it's very similar to that. But we're looking at multiple different things. We're looking at the outline. We're looking at the eyes. We're looking at the shape of the nose. We're looking at the shadow. We're looking at the age. We're looking at all of those things, and we're doing it instantaneously thanks to parallel processing. Okay, so basically, what we're doing is we are able to respond out of 100 faces within the fraction of maybe a few seconds. Our supercell clusters can take all of that parallel processing data and pick out grandma out of 100 different faces. And we can do it within the fraction of a second, or within at least a few seconds. Okay? All right, so ultimately, how do we see? Well, again, we have to take into consideration we're talking about two different theories of color blindness, or I'm sorry, of color vision, but we don't really know. We do know that we see in color, so that's still up for debate. But basically, light is going to reflect onto our retina, and it's going to trigger our neural impulses sent to our brain. That information, those impulses, are then have to be cataloged and decoded by the brain in the process of perception. Okay. So what we're doing is we're basically taking all of this data and we're turning it into an end result, which is why our brains actually don't always tell us 100% exactly what we're seeing, what we're smelling, what we're tasting, what we're hearing, what we're feeling. Okay, oftentimes it's our brain's desire to want to fill in the blanks, and sometimes our brains want to go the shortcut route. So if we see 3 or 4 pieces of data that link us to grandma's face, guess what? It's grandma. All right, we're going to automatically make that leap. Okay. All right. So again, just to kind of recap, the scene. If we're looking at a certain aspect, all right, we have the scene. Parallel or retinal processing is going to basically be the receptor rods and cones sending messages through the bipolar cells and then the ganglion cells. Feature detection. This is where we're breaking things down into features, edges, lines, angles, etc. Parallel processing. Is it moving? What color is it? Does it have depth? How far away is it? Etc. And then finally, recognition. This is the perception. This is where we're like, oh, yes, I see that is grandma. Okay, folks, that does it for 18. A little bit longer than 17, but thanks for bearing with. Make sure you know the parts of the eye. Make sure you know rods, cones, optic nerve, and blind spot, not to mention the retina, the fovea, etc. The 2 theories on color sight. Make sure you know feature detectors and what they do, and parallel processing and why it is important. All right, folks, that does it for 18. Stay tuned for the rest of Unit 3 coming up shortly.